Alright, we're going to continue our discussion of glycolysis in this video. Last video we covered the energy investment step and the lysis step of glycolysis. And at the end we ended up producing 2 molecules of glyceraldehyde 3-phosphate, also known as G3P. So in this step what we're going to try to do is we're going to try to capture some energy in the form known as ATP. Now, in your textbook, in the appendix, starting around appendix page 5, you have a detailed outline of the events that take place in glycolysis so that you can see off of these carbons that I've drawn here as circles, there's other atoms that are attached. One of the atoms that are attached right here off of this first carbon is a hydrogen atom. Now, one of the first things we want to do in this energy capture step is we want to be able to bring in an inorganic phosphate and I want to be able to put it right there where the hydrogen is. But working with these, it's kind of like working with Tinker Toys. Imagine this carbon being a little brown piece of a Tinker Toy, and I want to put this phosphate there. But before I can put this phosphate there, I have to remove this out of this slot so that I can put it there. So what we're going to do is we're going to remove this hydrogen and we're going to pass it to an electron carrier known as nicotine adenine dinucleotide, and we're going to reduce that electron carrier to form NADH. And in that hydrogen's place, I'm going to put that inorganic phosphate. We're going to do that to the same glyceraldehyde 3-phosphate molecule here. Now I want you to take a look for just a moment at the difference between this bond and this bond. This is a regular covalent bond here. This is a high energy bond between this phosphate and this carbon. In order to be able to phosphorylate adenosine diphosphate to make ATP, we're going to have to put a phosphate that is charged up against another charged phosphate. Like charges tend to repel one another, so it's going to take a significant amount of energy to be able to accomplish the phosphorylation of ADP. Now that we have this high energy bond, we're going to be able to do that. So for each of these molecules, we are going to bring in an ADP, and we are going to phosphorylate with this phosphate to form ATP. This is what we call substrate level phosphorylation because we're moving a phosphate from one molecule, one substrate, to another to phosphorylate it. So we do the same thing here, producing another molecule of ATP. I'm going to get rid of these because they're not shown in your drawings in the textbook. When we're finished with this step, this is the molecule that is represented in your textbook in chapter 5. A lot of students will think that these two are the same. These are not. This is glyceraldehyde 3-phosphate, whereas this one is 3-phosphoglyceric acid. Remember, we moved a hydrogen off of this one up here and gave it to this electron carrier, and that's no longer associated with this molecule. Now we're not going to be able to stick another phosphate here, so we're going to have to work with this phosphate to try and get it in a high energy orientation just like this one. So what we're going to do is in 2 steps for both of these molecules, we're going to take out water from each of them. So remove 2 hydrogens and an oxygen from each one, forming water. And we're then going to move the phosphate to the central carbon. It's kind of crowded. Remember, it's charged, so it doesn't really want to be crowded. And so when we put it on that central carbon, we move it into a high energy orientation. And just like up here when we had a high energy phosphate, we were able to make ADP. We're going to do the same thing down here. We are going to bring in a molecule of ADP to each one of these. And we're going to produce a molecule of ATP. That leaves us with 2 3-carbon molecules. And those 3 carbon molecules, this is the final product of glycolysis. We call it pyruvic acid. You'll also see it referred to as pyruvate. So your final carbon product in all of this process, after all 10 steps of glycolysis, you produce this pyruvate. Other things to take a look at after going through this whole thing. Recognize that you started off by investing 2 ATP. So our initial investment was 2 ATP. Now let's look at the total that we produced. We produced 2 here and 2 more here. So we produced a total of 4 ATP. But if we subtract our investment out, we only get a net gain of 2 ATP to run things in the cell. So most organisms that have the necessary enzymes or the structure that's needed to perform the next phases will go ahead and perform cell respiration so that they can get some additional ATP. And what they're going to be doing, I pointed out at the first video that we're going to start to try to remove hydrogens from glucose. We only removed 2 hydrogens of those 12 and passed them to electron carriers in this particular phase. Now these NADH that I've circled here, these are important molecules. These are like little paychecks. We're going to move these over to the 3rd phase in cell respiration and cash these in to produce more ATP. So we don't neglect these. These 2 NADH that we produce here in glycolysis are valuable. Now, we start off, glucose is C6H12O6. We removed 2 of those here, so we still have 10 hydrogen that are left that are associated with these pyruvate. So we want to take these pyruvate molecules and we want to try and capture the rest of the hydrogen onto electron carriers as we move into the next step called the citric acid cycle. So the citric acid cycle is our next focus as we discuss carbohydrate catabolism.